Hello, and we are going to talk today about proteins. This is our third organic compound in living cells. Here, if we remember, we talked about carbohydrates and lipids. Now we're going to look at proteins. Protein function is very diverse. There are a lot of functions that proteins serve in cells. They are structural components of cells. They send messages and receive messages between cells. They, as far as structural components and enabling molecules to pass through the phospholipid bilayer, you already know that proteins function in that capacity as transport proteins. They also play roles in defense against diseases, make up skin, hair, muscles, and skeleton parts. But one of the most important roles that proteins play is as enzymes. You've probably heard that word before, but enzymes are molecules that assist in chemical reactions. Enzymes are proteins. And without enzymes, there are thousands upon thousands of chemical reactions in our bodies that would not occur. The enzymes make them happen. So the protein function that is probably one of the most critical is as enzymes. So let's look at what the building blocks of proteins are. What is their structure? Proteins are made out of amino acids. That's the fundamental unit that makes up proteins. What is an amino acid? Well, right here we have an example. This is the chemical structure of an amino acid. An amino acid has an amino group. It looks like this. It has a nitrogen and 3 hydrogens. It has a hydrogen right here and a carboxyl group. You may also have heard this referred to as a carboxylic acid. That's the acid group right there. And right here, an amino acid has what we call an R group. R does not stand for an element on the periodic table. It stands for some variable group that has a number of carbons on it, probably some hydrogens and oxygens as well. This group varies from amino acid to amino acid. Depending on what that R group is, will tell us which specific amino acid this will be. We have various amino acids that bond together to form what we call polypeptides. When amino acids bond, the bond that is formed, and if you can see right here is an example. Here's an amino acid and here's another amino acid. When they bond, this nitrogen bonds to this carbon and it forms what we call a peptide bond. So when 2 amino acids bond and form this peptide bond in between them, we get a dipeptide. It is simply a polypeptide that only has 2. Di is for 2 amino acids that are bonded together. Protein molecules range anywhere from 50 to 3,000 amino acid units that are bonded together. So they can get very, very large. This is a tiny, tiny little example here. The type of amino acids, so which amino acids they are, the number of them, how many of the amino acids are strung together, and the sequence, what order those amino acids are bonded in, those determine the protein. So it's very important which amino acids are bonded together and in what order and how many of them there are. Another item to note is that as far as the R groups, some R groups are polar and some R groups are nonpolar. So some proteins will dissolve in water and others do not. And that's just something to note. So now let's look at the overall protein structure. We've learned what the individual components are that make up our string of amino acid chain. What we call that, here is what that is representing. Here are our amino acids bonded together with those peptide bonds, right? This right here that we've just talked about is simply called the primary structure, or one with a little prime after it. The primary structure of a protein is the sequence of those amino acids in the polypeptide chain. But proteins are much more complex than that, much more complicated. Once we have our primary structure, that protein will then undergo a folding, if you will, into its secondary structure. It folds and it twists. Two of the major formations that happen here are alpha helices and beta sheets. This right here is an alpha helix. This is a beta sheet. You can just simply see it looks like a wavy sheet, and this is a coil. It coils around. It's a helical coil. So these are secondary structures. Once we get this amino acid chain, it will fold into these structures. Then the protein will undergo a conformation where it has more complex folding. It's called the tertiary structure, or a 3' for short. This will determine a protein's function. And it basically just folds into a more complicated design that enables it to function in a very, very specific way, almost like a lock and key. Here you can see there's a little space in this particular protein where some type of other enzyme or molecule may fit, and it would be able to facilitate some chemical reaction. So the tertiary structure will determine its function and what it really does in life as a protein. The quaternary structure, or 4', is the structure that not all proteins will undergo this, but when we have two or more tertiary structures that combine together, it will form the quaternary structure. Each protein has a very unique shape and therefore a very specific function, and these primary, secondary, tertiary, and quaternary structures are some intricate and beautiful designs that proteins undergo in order to become active and functioning. Advanced ideas. Research specific proteins and their functions. What are their structures? Why are they able to carry out their specific functions? I look forward to seeing what you come up with, and I'll see you in class.